Today we are starting with the last session of the course, that is biotransformation. So I hope that after this we'll have one last lecture again, which should cover the very last thing which we should have be doing in this course, so that by the end of next week we would have finished with everything in MLB 203 Biochemistry 2. I understand maybe after next week it means you'll be having 2 weeks to exam, and so then we would have succeeded in our plan to have at least 2 weeks before exams. If there is a need for maybe revision, maybe we can do one lecture for revision. I don't know how that is going to be, because it's a lot of things. So if you want to do a revision, one lecture, it will be such a rush. I don't know if the need will be, but well, that is up to you. If you decide that we have a revision lecture, you let me know. Then that is after next week, and then we can have a quick review of everything which we did over the semester. Then you can have the rest of the time to prepare yourselves towards the exam. So diving into biotransformation, we talk of pharmacology if we are referring to substances of medicinal property. We basically divide this field of study into two major parts, which is pharmacokinetics and pharmacodynamics. I mean, for you as medical laboratory science students, we look at it this way as pharmacokinetics, pharmacodynamics. But if you were doing toxicology, for instance, similar things that we'll be doing, but we will then be referring to, for instance, the pharmacology is toxicology. So instead of pharmacokinetics and pharmacodynamics, we would be talking about toxicokinetics and toxicodynamics. That is to say that the focus is going to be on the toxicity of the substances that we are studying. But in this case, our focus is on the therapeutic properties of the substance, if we are looking at it in terms of medications. Now, pharmacokinetics simply deals with what the body does to a xenobiotic, a substance that is being taken into the body. Just like if you take in food, you talk of digestion, which is the food goes through the digestive system, and you describe that process of the movement of the food from the mouth through the esophagus to the stomach, and then all the other things. You describe all those processes of digestion. Similarly, when you take in a chemical substance, like a compound, a medication, a chemical substance will undergo some metabolic processes. And mainly these metabolic processes occur in the liver. But we do have 4 major stages that these substances pass through, what we refer to as absorption, distribution, metabolism, which actually is the term that is used to name that last part of the course. Metabolism is actually biotransformation. And then excretion, which is to say that biotransformation is an aspect of pharmacokinetics. So then you have absorption relating to how the substance gets into the body, distribution, how the substance gets to all the parts of the body after the drug is taken in, how it gets to all the tissues, all the organs. Metabolism, you will talk about the changes that occur to the substance that have gone into the body. And then finally, excretion through different routes. You have excretion of the substance out of the body. Pharmacodynamics, on the other hand, we'll be talking about the effect of the drug on the body. So this is where we talk about, mechanisms of action of the drug. At the cellular level, we're talking about drug receptor interactions, just like what we did with, cellular communication. We talk about ligands having interaction with receptors on the surface of the cell. Similarly, we have drug receptor interactions where the drug will be the ligand and the receptor will be a biomolecule which is embedded in the cell membrane. Well, some drugs can also penetrate through the cell membrane into the cell to now interact with receptors inside the cell. There too, we talk about dose response relationships. That is to mainly talk about concentration of the drug related to its therapeutic effect as well as its toxic effects. At what concentrations will the drug, be of a therapeutic, be giving some therapeutic effect, or at what concentrations will it be giving some toxic effects? So generally, that is what you find when you buy any medicine and you open it, you look inside, the leaflet, you see that mainly the things which are written there are divided into these two major parts, pharmacokinetics and pharmacodynamics. So by the time we finish this, the next time when you take a drug and you open it, you look at the leaflet and you are reading through, I hope you have a better understanding of the things that are written in those leaflets, talking about mechanism of action of the drug. And when they talk about absorption or distribution, or when they mention some metabolic effects or how the drug is excreted, you should have a better understanding. Right. So if you have an exposure to any substance, but let's limit it to, say, medications, the absorption of that drug is okay. Somebody is saying that he can't hear. Is it only that person who cannot hear me, or everybody else cannot hear? If there's anyone in the group who can hear me, the person can write yes. So he writes in the chat, yes, 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 so that I know that perhaps it's only that person that is having an issue, maybe with the network. All right, so Benjamin, okay, no, I mean Benjamin Ejeba, there are so many people who can hear me, so I think the issue might be with your network, because so many are writing yes, yes, who can hear me. So you kindly check your network. All right, so absorption refers to how the drug is introduced into the body, which we'll take a look at, the different ways that drugs get into the body. And then after the drug gets into the body, it gets into the bloodstream, gets distributed to all organs, to all tissues, and okay, so Benjamin, okay, somebody is advising Bar to check his network or maybe his system. That's so that he can. Okay, he says he can hear now. Good. All right. So after I have the drug getting distributed, you can have the drug, binding to perhaps in the bloodstream. You can bind to some blood proteins like albumin, for instance, or maybe even the tissues. We can bind to some tissue cells before it will have an effect, whether it is a therapeutic effect or it's a toxic effect that it will be having there inside the organism. Now, from distribution, you can either straight away have an effect of that drug which is introduced into the body, as once it gets distributed to the tissues, gets to the cells, it can have direct effects, which is to say that the substance which is being introduced into the body itself has a therapeutic effect. Or you have from distribution a process of biotransformation, which means you have the metabolites of the drug, which will rather either be binding onto some tissues before it will have an effect, or the metabolite is what is having an effect. Well, in some cases, you have the drug itself having effects and some metabolites also being active metabolites and having effects. So you can have these situations where you have a drug, what is referred to as a prodrug, that a drug is taken in, and then that drug in itself is not having any effect, but it's metabolized. So the drug is taken in, and then it is metabolized, and then the metabolite is rather, having a therapeutic effect. So that's what we refer to as a prodrug. And then, of course, after over a period, after the drug gets metabolized, or even in unmetabolized form, that drug can get excreted out of the body through different pathways. All right, so we take absorption, because we started from, say, absorption, because we are looking at pharmacokinetics, that we saw that the various stages are absorption, distribution, metabolism, and excretion. So 4 stages. Usually this is referred to with some acronym, ADME, ADME, absorption, distribution, metabolism, and excretion, which are the 4 stages of pharmacokinetics. So actually today what we'll do is we'll be looking at pharmacokinetics, so that the next time we'll be looking at pharmacodynamics. So if you have any drug taken into the body, as you can see, the various pathways, maybe not exhaustive, but I think this is very exhaustive enough, because you have, the route of entry of the drug into the body can be oral, which is a very common pathway of drugs being taken in and administered. So through the oral routes, gets into the gastrointestinal tract. And once it gets into the gastrointestinal tract, just like the way food gets absorbed into the bloodstream in the small intestines, it gets absorbed into the bloodstream and starts flowing to all the organs and the tissues. And then in the liver, it is in the liver that you have metabolism of this drug. Then you have metabolites and part of the drug itself getting into, because the liver is helping to do what we refer to as detoxification. But when you take in any substance, what is the liver doing? The liver is trying to it's a xenobiotic. It's an external substance you have taken in. So the liver is trying to get that substance out of the body. And so sometimes you hear the term detoxification also being used for biotransformation, even though it is not in all cases that metabolism of a drug will result into detoxification. In fact, in some cases, the biotransformation of the drug will rather be resulting into toxication or toxification of the substance, which is to say the metabolite can be more toxic than the substance itself. Of course, you have the drug after it's metabolized now gets secreted into bile. We saw bile, I think, when we were doing jaundices. So you know where the bile is inside the liver. You have the gallbladder, which stores the bile, and that bile is then secreted into the digestive system, into the duodenum, and eventually will come out in stool. So that is one route by which you can have excretion of drugs out of the body through stool. You can have that. So when it is oral, to go through that part all the way and then get excreted through stool. Of course, part of it will also get excreted through other means. You can have inhalation, that is through the nose. Abdul Rahman, I think I was still on that third slide, so that's why I didn't say anything. Whatever it is, the video is getting, we are having the lecture being recorded, and if anything at all, after the lecture, if there is anything, if you ask, I can go over it again. You have inhalation. So drugs taken in through the nose will get into the lungs, into the small pockets inside the lungs, and then can get exhaled out of the body. Of course, even not only drugs, even if you are referring to some endogenous substances, that's I think I think your group, you didn't do metabolism of, say, maybe lipids, for instance, so that when you have the formation of ketone bodies and later some of these one of the ketone bodies is acetone, and that acetone can get exhaled through the nostrils. So you can have such an excretion pathway, which is by expiration. Now you have another common way of taking in drugs, intravenous, that's by injection, and this straight into the bloodstream, and this can also go into the lymph. Now from here, you can have the drug getting filtered out in the kidneys, and that will go into the bladder and get excreted through urine. In fact, the urine is a very major pathway through which you have a lot of excretion occurring of drugs. You also have the drug after in the blood. The blood, you are having cells as well as plasma. And so the extracellular fluid, which is the plasma, is also carrying some of this drug. And then you have some glands, exocrine glands, like the sweat gland under the skin, which will be responsible for excretion of the drug through sweat. So that is another pathway by which drugs get excreted out of the body through sweat. You can have some of these, after it gets to some cells and some organs, it gets excreted through. So when you say soft tissues, really, that's very broad because you are talking about the muscles, which eventually will end up either in the skin or through maybe other pathways like the mouth, the nose, even through the ears, maybe through wax and things like that. You can have such things occurring if you say so. That's very broad. If you say soft tissues. Now, if you say bones, yeah, as you can see, bones. So what you see is that there can be storage of some substances inside the bones, like some particularly toxic substances, like some um, if you have exposure to some herbicides, for instance, or some insecticides that are uh, lipophilic and can get um, dissolved in say fat cells, and some of these uh, triacylglycerol, you find it in the bone marrow, and you have some of these substances that get stored inside the bones. What then happens sometimes is I think I talked about this before in your class, that sometime in old age or when you are sick and you start to lose weight and some of these uh, toxic substances begin to uh, now get back into the bloodstream. It is then that the toxic effect will begin to uh, to be manifested. So you can have the drug, some getting stored inside the bones. Then of course, yeah, I just talk about this last one, which is the adipose tissue. So this adipose tissue is under the skin, where you can have, say, lipophilic drug substances getting stored. And you can see from that adipose tissue, that adipose tissue, when it breaks down, it will go back into that extracellular fluid. So that's just what I was describing, that if you have some toxic substances that are lipophilic and they get stored in the adipose tissue, as if they are not there, later in life, maybe in old age, when you start to lose weight, these substances then get dissolved back into the bloodstream, and the toxic effect is felt afterwards. So I remember one of my students was telling me a story about a dad who was diagnosed of having some pesticide intoxication. I mean, when he was old already, like it wasn't that he was exposed at the time and then he was seen, but so it was sort of a surprise that, say, he was having such uh, a pesticide intoxication. It was in one of these classes I was explaining to them. That's when she said, okay, so perhaps that's what may have happened to the dad. That maybe at a younger age he was being exposed to these things. Sometimes, maybe because of your work, you are getting exposed to some of these things and they are getting accumulated. And it is later in life that the effects will then begin to be manifested. Right. Now, we take the oral administration and look at a typical, dosage of drugs through oral administration is such that, you are told, take this maybe after every 4 hours, or you are told take this after meals in the morning, in the afternoon, in the evening, which is to say that you are taking a drug with repeated doses. That is usually how you take these medicines. Not is not usually just a single dose. Now, what you see on the screen is a concentration curve against time, that is if the drug is taken in by oral administration. We're looking at a concentration of the drug substance in the blood over a period of time. So this is looking at it over a period of 96 hours. Now, you see that at the time that the drug is taken, which is time 0 hours, see the concentration of the drug is 0 in the blood. And then the concentration begins to increase gradually until it gets to the maximum. Now, this portion of increase of concentration of the drug in the blood represents the absorption stage of the drug. Because it's oral. When you take the drug in orally, it will first get into the digestive system. And it is from the digestive system then that is going to get absorbed into the bloodstream. And so that absorption is gradual until you reach a maximum of that, the concentration of the drug in the blood. From there on, there will be a reduction of the drug substance in the blood. And that phase where you are seeing that there is now a reduction of the drug substance all the way coming down, represents, say, a biotransformation stage. And at a point, somewhere below where you see there is even much more decline in the concentration of the drug, such a phase represents excretion of the drug. Because what happens is that when you take the drug in and it's getting absorbed, mind you, there's distribution. So when it's getting distributed, it will get to the liver where it's getting metabolized, or biotransformation is occurring. So once biotransformation is occurring, it means the concentration of the drug is going to be reduced. And it will be reducing gradually until also the period of excretion, where now some of the drug is getting excreted through urine or through the other pathways that we talked about, until finally the whole drug is excreted out of the body. And you see in repeated doses, after that first curve that we've seen, you can see that if, let's say, after 24 hours, the drug is taken again, you see that absorption occurring again. After another 24 hours, the drug is taken again, you see there is that, again, absorption and then decline. So which is to say that, the dosage regimes are actually, determined with curves of this nature, that if the drug is given to, some, if you do clinical trials and to monitor the concentration of the drug over a period of time, you'll be able to tell the half-life of the drug, which is at what time does the concentration of the drug gets to, half of its maximum concentration. So you can have that to now determine after how many hours should the dose be repeated. That's how come you can be told either you should take a drug, one in the morning, one in the evening, or you are told to take it 3 times in a day, or you are told to take it once a day. So it depends on the half-life of the drug that you'll be giving such dosage regimes. Another thing too is that if you look at these repeated dosage curves, there is an average there. So you see there's an average which is in the middle. That is just to show the average of the therapeutic concentration of the drug, the concentration at which the drug will have a therapeutic effect. Now, you see at the peak will be the maximum, of course. The peak is the maximum, and then it begins to come down. And you also have the minimum therapeutic concentration there. So that is shown as C minimum. You see that also there. Now, what we can say about this is that the space between the minimum therapeutic concentration and the maximum therapeutic concentration, that distance gives you what we refer to as a safety corridor, which is to say that it is within that concentration range that you can have a therapeutic effect of a drug. If you have the concentration above the maximum, you may be getting into a concentration that will rather be having a toxic effect. And if it is below, then it won't be having the intended therapeutic effects. It's there, but it will just get excreted out of the body with such low concentration. So the concentration is important for substance to have its therapeutic effects. If it was toxic effect too, it's the same thing, which is why a lot of times those who study toxicology, well, they have a mantra to say that the dosage determines the toxicity. Because the substance which is toxic, if it's not taken over a certain threshold, will not have a certain toxic effect. The same way, a substance which is having a therapeutic effect, if it's not taken within a certain concentration, it will not manifest its therapeutic effect. So the concentration or the dosage is important to achieve the therapeutic effect of the drug. Right, so that is absorption by oral administration. Another common way you take in drugs is by the intravenous administration. And so here you see, right at the beginning, you can see the curve is coming from top down. And that's because injection, intravenous, goes straight into the bloodstream. And so right at the beginning, the concentration of the drug is high. Then it gets to, get distributed to all the tissues and the organs. And then later you see that concentration declines, much, much more, until all the drug is eliminated out of the body. And so that phase is what we refer to as the elimination phase. And where it is getting reduced so steeply, we refer to that as a distribution phase. So that's the difference you can have between the drug you are taking in orally and a drug that you are taking in intravenously. Of course, this also will be for drugs that you are taking in by inhalation, because when you inhale a drug, it goes into the lungs, and in the lungs it gets absorbed into the bloodstream directly in the alveoli. And so, in fact, one of the fastest ways to get drug substances into the bloodstream is by the inhalation process. It's also really, really fast. All right. Now, distribution. After you finish all that absorption, you have the drug now getting distributed to all tissues, to all organs. Now, major things you talk about in terms of distribution of drugs is volume of distribution. That is a theoretical volume in which, like a volume of a solvent, in which when you dissolve the drug, it is equivalent to the volume of fluid in which the drug is getting dissolved to give you the concentration of the drug in the body. I mean, this is to see how much of the drug will be in, say, plasma, and how much of it is going to be bound in, in the tissues. So usually you have the volume of distribution being a ratio of the concentration of the drug in the tissues against the concentration of the drug in plasma. If you have this ratio, the value is higher, or let's say above 1, then it means that you have the drug being more in the tissues than in plasma. But if you have the value, being lesser than 1, then of course that drug is more in plasma than it is bound to the tissues, because it's a ratio. So if you have the denominator, being higher, which is the plasma concentration of the drug, then of course you are going to have a lower value, which means that the drug is more in plasma than it is in the tissues. Now, similarly, you talk of binding of the drug substance to proteins, like the blood proteins, like the albumin. And this is where you talk of the term of bioavailability, because when you take in a drug substance, some of that drug substance gets bound and some of it is free moving. And indeed, it is the unbound free moving substance which is active. The bound is inactive. I mean, theoretically, that is how it's being looked at, because it is the free substance that can get to a target to have its effect. Once it's bound, it means it has done its job already. Now, either that is if it is bound to a receptor of its targets. But if it's just bound to, maybe like a blood protein and moving about, is bound, like conjugated. Something like when we were looking at the endogenous substance bilirubin, you saw, the process where it got bound to bilirubin, for instance. Then, of course, yes, you also talk about tissue binding. I think we've said much about this already, which has some impact on the half-life of the drug. If you have a drug being bound more to tissues than is moving in the bloodstream, then of course the half-life is going to be much, much longer. Because if it's in the bloodstream moving, then it can get excreted much quicker when it gets to the liver. But if you have a lot of it getting bound, it means that the release of the substance, into the bloodstream is going to be much, much more gradual. And so the half-life is going to be much longer. Because even if you take the blood alone, not taking, let's say, the soft tissues in addition, the blood alone has components. You did hematology, and so you know that you have, different components of the blood. You have some of the blood which is in plasma, and you have the rest that are bound to the blood cells. And this is also important, for interpreting, pharmacokinetic data across different species. Right. So that is about drug distribution. Now, when you come to drug metabolism, which also we refer to as biotransformation, this is divided into 2 main phases. We usually refer to it as phase 1 and phase 2 of biotransformation. Now, the main purpose of biotransformation mostly is for the conversion of a drug substance, which is lipophilic to be hydrophilic, because as I mentioned a lot of times, the internal environment of the organism is aqueous. And so if you have a lipophilic substance in the body, that lipophilic substance should be made more hydrophilic so that it can move freely in the aqueous environment. So you can have the substance which is getting to the body, not even going through phase 1, but it goes directly to phase 2. And phase 2 consists of just conjugation reactions. We've seen some of these conjugation reactions when we were looking at some metabolic pathway of bilirubin. We saw conjugation there. We'll look at it again so that you can remind yourself of that process of conjugation with glucuronic acid. And once it's conjugated, it gets easily excreted out of the body. Or you have the xenobiotic undergoing phase 1 metabolism before undergoing again the phase 2 metabolism. And phase 1 metabolism can result into the formation of metabolites, which is active. So you have an active metabolite, but of course its form is changed. It's a metabolite. It's no longer the original substance, but it's an active metabolite. Or you have a non-active metabolite, which means the metabolite will not have the therapeutic effect that the original substance is having. So you can have these two things occurring, active or non-active metabolites. And each of these can undergo conjugation, which is the second phase. So you have a drug substance that undergoes phase 1 and then phase 2 before it gets eliminated. Or you have drug substances that are undergoing directly just phase 2 and then they get eliminated out of the body. So you can have these situations occurring with drug substances that are entering the body. Now, what are these phase 1 and phase 2 reactions? Now, phase 1 reactions are mainly 3 types of reactions, which are oxidation and reduction reactions. And the third is hydrolytic reactions. And most of these reactions, they are occurring in the endoplasmic reticulum or in the cytosol. So that's where you find these reactions occurring. Under oxidation, you will see reactions like hydroxylation, decarboxylation, formation of oxides, desulfurization, that is if you have some sulfate groups inside a drug substance, the removal of them, dehalogenation, oxidation of alcohols, oxidation of aldehydes. These are oxidation reactions. And then reduction reactions will be reduction of some aldehydes, azo reduction, that is to say reduction on nitrogen inside a nitrogen containing compound. So you have those. Hydrolysis, splitting of the molecules into 2. So it can be breakdown of amide bonds or breakdown of some ester bonds. So, usually, I think if you look at, I don't know if we saw, I think when we were looking at hormones, we did see a process of lipolysis that we referred to the breakdown of triacylglycerol into fatty acids and glycerol, and those were ester bonds which were broken between the glycerol backbone and the fatty acids. So that's splitting of esters. So you can use that as an example as far as hydrolysis is concerned, that is splitting of esters to refer to maybe a drug substance which is some having a lipid nature. Well, we have some examples, some specific examples we'll look at. I hope we'll see some hydrolytic processes there. Then the phase 2 reactions. they are conjugation reactions. And conjugation is about, joining of two or more molecules. And I mentioned earlier that what it is for is to make a substance more hydrophilic, so that you see a hydrophilic compound which is binding onto a hydrophobic compound to make it more hydrophilic, sort of being a carrier of that hydrophobic substance to carry through the aqueous environment. And so you see, I think we've seen this with bilirubin. That's conjugation with glucuronic acid. We've seen that. We see some conjugation of sulfates. I think we'll see that example with some medicines. Acetylation, so conjugation with an acetyl group. Glutathione conjugation. I think we talked about glutathione with maybe not in your class, because we didn't do metabolism of carbohydrates looking at the pentose phosphate pathway. But I think you must have done it in first year, because it's in first year you did those metabolic pathways. Then methylation. We saw some form of methylation when we're doing I think the nucleic acids. Methylation only means addition of methyl group. So you can see all of these, they are all conjugation reactions. Phase 2 reactions are all conjugation. It's an attachment of a residue on a molecule. All right. Very good. So now we are seeing the very first example, which is what we saw earlier when we're studying hemoglobin metabolism. And here you can, so here what you can refer to this as a biotransformation of an endogenous substance. It's not a xenobiotic. It's not an external substance which is coming in, but this is endogenous. The bilirubin is being produced inside the organism itself, and due to the metabolic processes that hemoglobin undergoes, there is that breakdown of hemoglobin. After you have breakdown of the red blood cells, you have hemoglobin breaking down. We saw that cycle of the breakdown of the red blood cell after some number of days, 120 days, when the red blood cell is mature. So what you see in that process, say in the liver, even first of all in the blood, you can see bilirubin was bound to albumin. And I talk of it as a conjugation reaction there. But in that case, it's also to make it hydrophilic, so it can move freely in blood. And then when it comes into the liver, you are seeing another conjugation where now you have a glucuronide, because then after it moves from blood, only bilirubin is carried into the liver. The albumin is left in the blood, and that bilirubin is then conjugated on diglucuronide. That's what you see here, the glucuronic acid binding onto the bilirubin, which was broken down. And so you now have that bilirubin diglucuronide. So the bilirubin diglucuronide, you can see, moves from the liver now into the bile. That is, it will get stored in the gallbladder and then now be excreted through the bile duct into the digestive system. So we saw these things. And you can even see clearly that there's even excretion here of that endogenous substance. So this is bilirubin excretion, either in urine or in stool. So you can see all of these things happening with bilirubin. over here as an endogenous substance. All right, I think we'll take some specific examples with drug substances and some of the things that we have talked about so far. And the first we are looking at is oxidation of alcohols and aldehydes. And here you see ethanol is getting oxidized. So pay attention to the hydroxy group, which is on ethanol. And you see that hydroxy group on this ethanol, when it's oxidized, you now have on that second carbon, that carbon which was having CH2OH, and that hydroxy group now becomes a double bond O. So you now have an aldehyde. That's an acetaldehyde. And you have an enzyme, the alcohol dehydrogenase, ADH, catalyzing that reaction. It's an oxidation reaction there. And that reaction can even further continue where you have acetaldehyde dehydrogenase. And that acetaldehyde oxidizes the acetaldehyde to acetate or acetic acid. So that is further oxidation. You now see that the aldehyde group is now oxidized to a carboxylic acid group, the COOH. It's a carboxyl group. So that is oxidation of alcohols and aldehydes. So it's a reaction you see with these functional groups. Let's say these hydroxyl groups or an aldehyde group in a drug substance. These oxidation processes can occur on those substances. And of course, all the enzymes that carry out some of these reactions mostly are in the group of the enzymes we referred to much earlier, the cytochrome P450 enzymes in the liver. When we were doing hemoglobin, we refer to those enzymes as hemoproteins, if you remember. So many of those enzymes are oxidoreductases. They are involved in oxidation and reduction reactions in drug metabolism. Good. Now we take a specific example of a painkiller, pain reliever, antipyrine. I mean, this is intentional. I brought this so that we can see like different types of reactions occurring at the same time on one molecule. One molecule can get converted into different metabolites. So it's not like when a drug substance is taken, it gets converted into only one type of metabolite. No, one drug can get metabolized into different compounds. And this an analgesic. So we start from the very first stop and take the arrow one by one, and intentionally too, I have shaded specific groups that are getting converted so that you can easily relate. So the number one, that is the antipyrine itself, which is at the top. And pay attention to the groups that I have shaded. Shaded this group more like pink or violet, light violet, and then this is green. These methyl groups. This is on N2, and this is on carbon 3. I mean, the nitrogen, which is binding to the ring below it, there is N1. And so if you count clockwise, the second nitrogen there is N2. The carbon there is C3, then C4 that way. All right. Now let's see. So the very first arrow here. Let's see what is happening. See on that CH3, you are having CH2OH. CH2OH. That's an oxidation reaction because one hydrogen is gone. And then you have a hydroxy group coming in place of it. It's an oxidation reaction. All right. Now, or maybe sometimes maybe you can refer to it as a hydroxylation if you want to say it's a hydroxyl group, which is but hydroxylation is an example of oxidation. And if you look at the slides, I think 2 slides before this, where we did that grouping of the reaction types, you saw that hydroxylation is one of the types of oxidation reaction. So it's oxidation, but specifically you can say it is hydroxylation. Of course, that compound, the number 2, look at the name at the side. 3-hydroxymethyl antipyrine. So clearly, it's a hydroxyl group that has been attached there. Now see, the second arrow here, and you see the CH3, which is on N2, shaded green. You see it's changed, and you now have just a hydrogen there. So that reaction, that second arrow there, that reaction there is dealkylation. You can say it is N-dealkylation because you are having a removal of an alkyl group. That residue, that's a methyl group. So if you want to specifically mention that it's a methyl group which is removed, then it means it is N-demethylation, removal of that methyl group. But if you want to refer to it as, a general, removal of an alkyl, that's say an alkane residue. Alkyl will be an N-dealkylation to give you that compound number 3, which is norantipyrine. Now, the next arrow, here you see the compound 4, which is formed. I've shaded the hydroxyl group on it there. And you can see the original compound antipyrine is not having any group on that carbon there, carbon 4. There's nothing there. But here you have a hydroxyl group. So that is another hydroxylation reaction, which is an example of an oxidation reaction. And the compound there, number 4, what's the name? 4-hydroxyantipyrine, because the hydroxyl group is on carbon number 4. And then the arrow, the last 4 arrow, which is the last 4th arrow at the top there, the compound number 5, which is formed. There to have shaded this hydroxyl group on the second ring below. Because you see, there's no hydroxyl group there in the original compound. Number 1, you see, there's no hydroxyl group there. But here, there's a hydroxyl group. So that's another hydroxylation reaction. So another type of oxidation, hydroxylation has occurred to give you the compound number 5, 4-hydroxyantipyrine. See the difference between the number 4 and number 5. 4 says 4-hydroxyantipyrine, and 5, 4'. Yes, 4' because it's the second ring below. Because if you are counting that carbon in the second ring below, which is bonded on the nitrogen, that carbon is going to be carbon number 1. And if you continue to count clockwise, you have carbon 2, carbon 3, and so you have carbon 4 on which that hydroxyl group is. That is on the second ring, that's why it's 4'. And now you can even see that from here, what you see is that you are having both hydroxyl groups. Either you had this number 4, which was having a hydroxyl group here on carbon number 4, and then there is another hydroxylation to add this hydroxyl group to it. Or this last reaction here will give you the same thing if you have the hydroxyl group on the 4' first, and then you now have a hydroxyl group attached on this 4th carbon. So you can either have 4-hydroxy antipyrine, which will be converted to this last 4' dihydroxy antipyrine. Or you first have the 4' hydroxy antipyrine, which is then converted to now 4' dihydroxy antipyrine. And then the last one over here too, we see the shaded portions. This is much easier because they are just one after one below the other. So you can clearly see this was a CH2OH, which is now COOH, a carboxylic acid group there. So that is that's a carboxylic acid group. That's an oxidation reaction. So you now have a carboxy antipyrine for that compound number 6. This is to show, the different types of reactions which we refer to as the oxidation, reactions under the phase 1 reaction. So we can take another medicine and see, different. Okay, codeine. Codeine is another. I think those of you working in, pharmacies or even in the hospitals, you have come across some of these medicines. So we are just looking at, these metabolic processes, pathways that, the compounds that can be formed after the drug is taken into the body. Out of them, when they are getting metabolized, what can be formed out of them. So this is codeine. And here to have shaded this methyl group. And the conversion to norcodeine is just a removal of that methyl group. So when you look on that nitrogen, that methyl group is not there. And you see the process is N-dealkylation. Specifically, you can say N-demethylation. And then codeine is also getting converted here. And here too, you see that in place of this CH3, which is on this oxygen, here, you see, it's just a hydrogen there. And so that is O dealkylation. O, so it means the methyl group, that alkyl group was on oxygen. That's why it's now O dealkylation. But here, it was on nitrogen. And so it's N dealkylation. And you have that codeine converted to morphine. Morphine can also get converted to normorphine. And what do you see? Still N-dealkylation, where there's removal of this alkyl group from morphine. So when you look there, there's no alkyl group there. And so you have normorphine. Now, this is one of the examples of metabolism of a drug that will produce a more toxic substance than itself, like codeine getting converted to norcodeine. Norcodeine will be more toxic than this. Morphine is also going to be more toxic. Even normorphine will be more toxic than morphine. So you have that process rather can result into toxication or toxification rather than detoxification. All right. Oh, this is nice. This is a very common drug, which is paracetamol, metabolism of paracetamol. It's an interesting one. So you have paracetamol there. So that's the drug, acetaminophen. paracetamol. Now, what do we see? Let's look at it first, going upwards this way. That process there is glucuronidation. So that's a conjugation reaction. That's a phase 2 reaction. And what do you see? There is conjugation of acetaminophen or paracetamol to glucuronic acid. So that's glucuronic acid. That second ring, which is binding on there, that's glucuronic acid. So you now have, paracetamol glucuronide. In fact, you can see clearly that, let's say, 50 to 70% of paracetamol taken in goes through this pathway. I mean, that's to say that a lot of the drug just gets bound to glucuronic acid and then gets excreted out of the body, and you'll be left with just about some 50, 70%, some 50, let's say 30 to 50% of the drug will be left to undergo different pathways. You see that? So when drugs are taken in a lot, sometimes a lot of it will get excreted without doing anything. And it is just a minute amount that really has a therapeutic effect, as we'll see with paracetamol. Because here you see 25 to 35% of the drug can undergo another conjugation reaction, which is sulfation. So here you see you have the conjugation of a sulfate group on paracetamol there for you to get the paracetamol sulfate. So all these, they are non-active metabolites. This first one, paracetamol glucuronide, is non-active metabolite. Paracetamol sulfate is also a non-active metabolite to just get excreted out of the body. And you're having, so here, very good, we are seeing a cytochrome P, one of the cytochrome P450 enzymes involved in oxidation of paracetamol. About 5 to 15% of the drug gets oxidized into a benzoquinone. This is N-acetyl benzoquinone. And what you see in place of the hydroxyl group there, which was on the carbon number 4 there, you see that you have this cyclic ketone group. A quinone is formed. And in fact, this metabolite is actually a toxic metabolite of paracetamol. Even though it actually almost immediately, when it is formed, it almost immediately gets conjugated by glutathione and gets excreted. But if paracetamol is taken like overdose, it can result into paracetamol toxicity. And this actually is a substance that can result into such a toxicity of paracetamol that this, as you can see, can bind to protein and form some protein adducts, which can result into some serious liver problems, having bound to proteins that can, result into some internal clotting of blood and, some liver inflammation. It can actually result into fatality with paracetamol toxicity. This particular one, this benzoquinone, that's it's a toxic metabolite of paracetamol, which is responsible for paracetamol toxicity. And then the pathway which is below from paracetamol, here we see deacetylation. That is the removal of this acetyl group. there. So when the acetyl group is removed, you now just have a para-aminophenol. And that para-aminophenol is also another, non-toxic metabolite, but it is this para-aminophenol which gets bound onto arachidonic acid. And actually, it is this compound, these conjugates with arachidonic acid, this fatty acid bound onto this, which is referred to as AM404. This is actually the compound which is responsible for the analgesic effects of paracetamol. And what do you see? It's just 1 to 2% of paracetamol which goes through that pathway to come and give you this AM404. I mean, this arachidonyl aminophenol. That's what it is. All right. So that's a very common drug, paracetamol, that we've looked at. I think it's an interesting one there. We also can have another conjugation process, which is, acetylation. That is conjugation of an acetyl group. You'll find this with primary amines. I mean, compounds with amines at their terminal ends. And you see, so here you have this NH2, which is on this benzene ring. So that's an aromatic amine. And you have acetyl-CoA, from which you now have that acetyl group binding onto the amine group to give you an N-acetylated metabolite. I mean, you have some enzyme N-acetyltransferase that is responsible for catalyzing this reaction. And then, of course, you are left with a coenzyme there. I think when we are talking about some genetic polymorphisms, we can talk about N-acetyltransferase, which has some genetic differences in different individuals, responsible for either a person is a fast metabolizer or a slow metabolizer. I think getting to the end, we will see that very briefly. Now, elimination. So obviously what we've done is we look at the process of absorption, then we went on to look at distribution, and then we just finished looking at metabolism, which is the biotransformation of the drug into metabolites or different metabolites. Now we're looking at elimination. So the drug will get excreted either unchanged, in its unchanged form, not metabolized, it gets excreted, or it gets excreted as a metabolite, or both. And you have different pathways by which you have this excretion occurring. If it is through urine, we refer to that as renal excretion, because then the drug is getting filtered in the kidneys, and then will be excreted through urine. So you have that as renal excretion. If it is getting excreted through stool, that's a fecal excretion. Here you see either the drug is, when it's taken in orally, part of it, unmetabolized, unabsorbed, just binds to intestinal substances, the food, and just go down at the digestive pathway and get excreted in fecal matter. Or you can also have the metabolites, or the drug which is coming from bile. So you see that biliary excretion ends up also in fecal excretion. So you do have that. Or you have, so this is the biliary excretion, where you have the drug, or the metabolites from the liver getting stored in the gallbladder. and the gallbladder has bile, it then gets the bile to the duodenum, it has into the digestive system and can get with stool. Or if it's a gaseous metabolite or gaseous drug which gets into the lungs, it gets exhaled. So that's by expiration. By expiration, that the drug is excreted out of the body. So that is pulmonary excretion. So you can have such as well. You can also be having the drug getting excreted through the skin. That is with sweat, which is dermal excretion. So the sweat glands secrete sweat, and inside the sweat, you can have some of these substances also coming out with the sweat onto the skin in that process of dermal excretion. Or you have some drugs that can get excreted through tears coming from the eyes. That's lacrimal excretion. So you have the lacrimal gland secreting the tears onto the eyes. And even some of the tears can go through the nostrils coming down. So some of this lacrimal excretion can also be coming through the nose out, not only directly through the eyes, but even through the nose. You can have that also occurring. Because if you think of it, eye drops, ear drops, nasal drops, they all have a connection. So sometimes if you use eye drop or you use nasal drop, you realize that sometimes you feel the taste in your throat because all the canals are connected. The ear canal, the nasal canal, the buccal cavity, they are connected. So you have the tears which are being produced, in which you can have this drug getting excreted, also coming out through the nose. Then you also have salivary excretion. You can have some drugs taken in. Of course, this is a drug in the bloodstream, and these salivary glands are also some tissues that, you have blood flowing through them. And so you can have excretion of saliva containing, some amount of these drugs as well. So you have salivary excretion also as one of the pathways by which drugs are eliminated out of the body. You also have the mammary glands that can be secreting this, particularly in breastfeeding mothers. Which is why sometimes you see that some drugs, there's advice on it that not for lactating mothers, so that it will not be passed on to a child that is being breastfed at that moment. And sometimes if it's not clear whether the drug can get excreted in breast milk or not, it's just advised that the mother shouldn't take it at all. If it is known, if the concentration or that substance itself is not good for a child, then the lactating mother is prohibited from taking such a drug substance. All right. Now, that was much focus on the human body, but we can also look broadly also at situations where you actually will have biotransformation in the environment. So you have this substance, this 2,4,5-trichlorophenoacetic acid, is a herbicide which was used sometime in the 1940s, this substance was produced and was used as a herbicide. I think it was even this substance which was used even in the Vietnam War. The Americans used it to, they use like aircrafts to spray large fields, so that, the Vietnamese that were hiding under forest cover will no longer have, such a forest cover to be hiding. Only later it was realized that even though it was, sort of drying the field, killing the weeds so that the place will be bare, it undergoes this biotransformation in the environment, undergoing this hydrolytic process. And you see the acetic group here breaks away from it to have an acetic acid, and you are left with 2,4,5-trichlorophenol. And this 2,4,5-trichlorophenol can then get conjugated, two of them, to form some dioxins, which is the tetrachloro-dibenzodioxin. And this is a toxic substance. It's persistent in the environment. It doesn't get degraded that easily, and it's toxic, to mammals. And so because of that, this herbicide, this trichlorophenoacetic acid, has been banned from use because of this. And here you see biotransformation is occurring, not in a biological system, but it is so we can't even refer to it as a biotransformation per se. We can only say it's transformation, because if we say biotransformation, it's more like it's occurring inside a biological system, but it's occurring in the environment. That hydrolysis is occurring, and then you also have conjugation, because when you have the two molecules now binding together, you now have the TCDD. All right. I don't know if I should refer to this as a splitting of esters. It's not an ester per se. If the double bond was on this carbon, then that would have been an ester, but a double bond is one carbon away from it. So it's hydrolytic, but not a splitting of ester. Okay, then just another one, parathion. Parathion was also used as an insecticide. It's a nerve agent. It acts on insects as an inhibitor to the enzyme acetylcholine esterase, which is involved in muscle contraction and relaxation, which is to say that this parathion, when it gets exposed to light, undergoes photooxidation, to form para-ozone. In fact, para-ozone is more toxic, even to mammals, than the parathion itself. So, similar effect of a blockade of N-acetyl, the N-acetyl, how do I say, the blockage of the acetylcholine esterase, which is to do with the inhibition of the nervous system. Same thing can happen to a mammal, which means that it can cause the effect of paralysis. I mean, that is serious. And so this tool has been banned. It's not being used any longer. And you have also the process of hydrolysis, which is breaking off this phosphor sulfate for the phosphosulfate group there to give you a diethyl phosphorothioate. Is cleaved off. So that is also another hydrolytic reaction which is occurring there. And then at the same time, you're having, so that's all. You are just having the nitro group there. So you have a para-nitrophenol. So in place of that whole group, which is cleaved off. You just have a hydroxyl group there. So that's a hydrolysis. And then the horizontal pathway is giving you a photooxidation. So that's some kind of a conversion of this substance in the environment, which is, looks just like it's a transformation. It's a transformation of these substances by its occurring in the environment. It's relevant for you to take a look at this because you begin to appreciate the fact that it is not only when a substance is taken into the body that the substance can have, serious effects, like if it undergoes metabolism in the body to change into these toxic substances. But even when it is exposed in the environment and it undergoes this transformation in the environment, it can have such adverse effects on the body. And so, for instance, you see, we just talked about these dioxins, which are very persistent and really toxic to the body. And that dioxins are mostly produced from burning, which is why it's really serious that in Ghana, people just get up and be burning things. They just get up burning rubbish. Somebody just collects rubbish somewhere in front of his house. It starts burning and just polluting the whole area. Or you have a place like Agbogbloshie that people are just burning things. And it's like it's normal that everybody is looking at it as if it's just a normal thing. But it's really a dangerous thing to be doing, to be burning things and having these dioxins in the atmosphere. I think some of these things are resulting into so many disease conditions that it's just that because we don't keep so much of data, things we don't really know. Because TCDD can have serious effect on even pregnant women and have effect on the children that they give birth to. So it's a serious thing. Burning things in Ghana is a very serious menace that I don't know why, let's say, health professionals don't seem to see it as a very serious issue, just like what is happening now with the mining in the country with a lot of toxic substances. It is as serious just as that, the burning of things. Now we have phytosterol. This is a phytosterol is from the name it tells you clearly this is a sterol which is in a plant. You find phytosterol in nuts. There's this research work which talked about one of these water bodies, a river in the United States, one of the states that there was an observation of some fishes in the river that showed that there were changes in the female fishes. And so the female fishes started to develop masculine features and even putting up, masculine behaviors. The zoologists can study these things and get to know that this feature of this fish this is the male fish, this is the female fish. So they realize that the female fishes were developing male features and were actually getting sterile. And it was investigated until they realized that there was a factory along the river that was involved in processing of nuts. And they release the waste from the factory into the river. And the nuts that they were, nuts contain a lot of these sterols. So the nuts, that oil coming from it was having these phytosterols that when they get into the river, there were some microorganisms inside the river, some bacteria that were acting on these phytosterols. And by their enzymatic action on these phytosterols, they get them converted into these androgens. So as you can see from a phytosterol, you now have a testosterone, you have an androgen, it's a sterone. So that hydroxy group has been oxidized, to that cyclic ketone group. And you have also that side chain. Of course, even if the side chain is there, you have a oxidation of that hydroxy group. It becomes let's say instead of a sterol, you will now have a sterone. From a sterol, you have a sterone. So that resulted into the masculinization of the fishes. That's why you had these features manifesting in the fishes. So you see how transformation of a substance in the environment. This one particularly is biotransformation by microorganisms. That's biotransformation occurring there, and the kind of effect that it is having on organisms in the environment. Okay, Adam Hanna says, do we have any amount of water we use in taking medicine? Capsules or tablets. Are we not diluting the medicine? No, not really. You are not diluting it. The amount of water which you use to drink the medicine is not, going to have any effects on the concentration of the medicine. Mind you, when you take in the medicine is now going to get absorbed into the bloodstream. The volume of blood is not going to change because of the water that you are drinking. So that cannot have any effect on concentration of the drug. All right. Now another thing, okay, we can talk about this cimetidine. That's a drug which is used for the treatment of heartburns and some peptic ulcers. But when it's taken, it has some side effects. That's when it reduces gastric acidity, what happens is that it means that now it can result into conditions of bacteria and fungi in the gut to undergo, more proliferation. And in the presence of nitrates that are coming from food and water, edible things you've taken in, you will have the conversion of those nitrates by these bacteria and fungi into nitrates, into nitrites. And these nitrites can undergo further some bacterial, catalysis, converting these nitrites into nitrosamines. Because you have, together with some other compounds that are present in the digestive system, thiocyanate chlorides, and this, actually would increase, the concentration of nitroso compounds, and this further the action of fecal bacteria would then increase carcinogenicity of these nitrosamines. So actually, even though the drug is treating peptic ulcers and heartburn, it increases the risk of gastric cancer in the end because of the condition of a reduced gastric acidity that it creates. You then can have some of these serious side effects coming out of it, an adverse effect coming out of it, causing damage to the mucosal lining and having some inflammation to the digestive tract. All right, Collins Kofi Edu says, what about taking water after taking blood tonic? Won't the water dilute the blood tonic, limiting its effectiveness? I think this is the same question you are asking as Adam Hannah is talking about. You are talking about the blood tonic as if it is. I mean, you have to think of it this way: that the tonic that you are taking in, it's having some specific molecules inside it, some substances that are going to have their effect. When those substances are getting absorbed, they get absorbed into the bloodstream. So it's not as if the blood tonic is acting by contact on the intestinal tissues. That's not what it is. As if you, for instance, if it is an acid that you are pouring onto a substance and you want that acid to have some maybe corrosive effect or dissolution of something, and you dilute it, then you would have reduced the effect of it. That is activity by contact, but that is not what it is when you are taking drug substances. The drug substance gets absorbed into the bloodstream, and you have specific compounds, molecules that are inside what you are taking in that are going to interact with cells in the tissues to have their effect. All right, so I hope that makes it much clearer. So we're talking about, I think I mentioned this, that we also have things like genetic variations in biotransformation, which actually goes into what we refer to as personalized medicine, where the same drug which is given to different people, if it's given the same dosage, some people will metabolize it fast. And so once they metabolize the drug very fast, get excreted out of the body, then the therapeutic effect would have been reduced. Some people excrete this very slowly. And so what happens is if the same dosage regime is given to such a person, there will be accumulation of that drug, which will rather be causing some toxic effect in the end. So this is just an example of a study that showed the concentration of isoniazid. It's a drug which is an antibiotic used for the treatment of tuberculosis. And in different persons, let's say we use about 267 volunteers took this medicine, and you realize that you had the concentration of the drug in some persons high after 6 hours, and some others, somewhere intermediate, and then some others very low. And so you could see the distribution of this group of people into 3 different groups as slow metabolizers, intermediary metabolizers, and fast metabolizers, which clearly shows that if you actually do some diagnosis of these persons, perhaps in the beginning you need to give a certain dose of the drug to determine whether the person is a fast or slow metabolizer before you give that person a specific dosage. So it means the dosage for a person is customized, is personalized. It's not going to be the same for everybody, because if you give the same to everybody, you are going to be having different effects, which is actually a new area of medicine now, personalized medicine. All right. Seth, now, Kwadade says, can defect on the spleen have effect on... Oh, you are still on this sickle cell patient. After we finished sickle cell a long time ago, and now we are talking about biotransformation. You are back to sickle cell. How come? How come you are now talking about sickle cell? All right, so I will end here, and we have 20 minutes. So if you have questions, you can write in the chats. You can raise your hand. I will enable your microphone, and then you can speak.